We're going to go ahead and move on to our next speaker, Colby Hause. Colby is a second-year master's student in the animal biology program at UC Davis. She's been working with Andrew Ripel and Nan Fangi. Broadly, she uses telemetry as a tool for conservation science. Her current research aims to understand the environmental drivers of outmigration mortality by linking broad-scale habitat heterogeneity and survival dynamics of threatened spring-run Chinook salmon. Through improved understanding of the ecological processes that shape critical life history stages, she aims to promote science-based policies that support long-term conservation management of fragile anadromous fish populations. And with that, I'll turn it over to Colby.
Awesome. Thank you for that introduction. Thank you again. My name is Colby, and today I'm going to be talking about some work that we conducted in the spring of 2019, looking at outmigration survival of juvenile spring-run Chinook salmon in relation to physicochemical conditions in the San Joaquin River. So I don't think I need to go through too much more background, but I just want to emphasize that with recent efforts to restore the San Joaquin River back to conditions more favorable for native fishes, really central to that goal is this reintroduction of spring-run Chinook back into the San Joaquin to eventually restore a self-sustaining population in the river. And so this is where UC Davis really comes into play. So to evaluate the success of initial reintroduction efforts, UC Davis conducted telemetry studies, which were led by Dr. Gabriel Singer, where we estimated survival from the upper restored reaches of the San Joaquin River to the ocean in 2017 and 2018. And so these studies really shed light on overall outmigration success, which ended up being very low for each year, estimated at 2 to 5% in 2017 and less than 1% in 2018. But this research was really important in helping us identify spatial patterns in regional survival. For example, this high initial mortality after release through the restoration area, as well as high mortality through various pathways in the delta across both years. And so since we were able to identify these really clear spatial patterns in survival, going forward, we wanted to better understand some of the environmental conditions that were driving these patterns. So last year, our focal question was, how does spatial variation in river conditions shape regional mortality in the San Joaquin River? And so previous studies that have investigated similar questions have typically used temporally explicit models, which are really important for understanding the impacts of covariates that are highly variable through time, but are a bit limited in their ability to describe the effects of localized conditions on outmigration survival. And this is primarily because these models rely on data collected from fixed water quality monitoring stations. And if those are spaced far apart, they may not be able to detect these small scale changes in habitat. And this is evident in the San Joaquin River with the current array of water quality stations, which are very coarsely spaced and also inconsistent in the types of covariates that they are collecting data on, which further reduces the spatial resolution of those data. So to capture localized habitat variation across the San Joaquin River, we used a novel method of habitat data collection called the FLAME, which stands for Fast Limnological Automated Measurements. And this was a system that was originally designed by researchers at the University of Wisconsin for use in mapping high spatial resolution water chemistry data across large bodies of surface water. And so we put our own version of this FLAME concept in the spring of 2019. And this was a boat-mounted flow-through system that pumped river water up into 2 flow control tanks that housed a YSIXO and a Suna nitrate sensor, measuring a total of 8 water chemistry variables, which included temperature, pH, chlorophyll, conductivity, fluorescent dissolved organic matter, which is fDOM, turbidity, dissolved oxygen, and nitrates. And so while we were running at high speeds, we were able to record measurements at a rate of 1 measurement per second, and then simultaneously georeference each measurement with a GPS coordinate. So to relate habitat conditions, which we mapped with the FLAME system, to outmigration survival, we first deployed an array of over 100 acoustic receivers from the restoration area here up by A1, and this is the end of Reach 5, to the Golden Gate Bridge here at A17, and those receivers are indicated by the red points on the map. We then tagged 700 juvenile spring run Chinook salmon with 416 kilohertz JSATS acoustic transmitters and released fish in 2 different locations, half in the restoration area here at this upper green triangle, and then the second half in the delta at Durham Ferry here by A7. And then we ran 3 water quality transects over the course of the juvenile immigration window, 1 prior to fish releases and 2 during outmigration. And so now I'm going to go through some of our statistical methods for the environmental data and then talk about how we incorporated those data into our survival analysis. So after water quality data were post-processed, data across all 3 transects were summarized to obtain the mean and the coefficient of variation of each water chemistry variable at each GPS location and corresponding river kilometer. And so this ultimately resulted in 2 primary data sets that represented unique characteristics of the environmental data. The first data set reflected the variation in water quality variables across space, which was represented by the mean of each variable at each location. And then the second data set represented variation in these same variables, but across time, which was reflected by the coefficient of variation, or CV, of each variable at each location. And then to classify habitats along the San Joaquin River, we applied a agglomerative hierarchical cluster analysis to each data set. And essentially what this did was just form groups within each data set based on similarities in those data. And then we used principal component analysis to further reduce data dimensionality and identify some of the key variables that accounted for the majority of the variation that we saw in our data while minimizing loss of our original information. And so to jump into results, 2019 was considered a wet year. And to give you an idea of what flow was like in relation to our different sampling events, the plot on the left shows flow in cubic meters per second from the Mossdale CDET gauge station located at the star on the map. And the dotted line marks the timing of our upper fish release in the restoration area. The dashed line marks the timing of the delta fish release. And then the 3 solid vertical lines mark the timing of our flame transects. So 1 occurring prior to fish releases, the 2nd occurring after the delta release, and then the 3rd occurring 5 to 7 weeks into the outmigration of both groups. So cluster analysis on the mean data set resulted in 6 distinct regions that best accounted for spatial variation in our environmental data. So that's this panel here on the left. And then cluster analysis on the CV data set resulted in 3 distinct regions that best accounted for spatial differences in temporal variation in our environmental data. So for the survival component, we fit a multi-state market capture model to our data following methods described in Buchanan et al. 2013-2018. I'm not going to go into the weeds of this model, but if you have any questions about it, I'm happy to field those after the presentation. And so we then constructed 4 candidate models, each with different parameterizations of reach-specific survival to represent competing hypotheses on the relationship between survival and habitat conditions. And so models M0 and M1 did not take any environmental data into account, and they modeled survival as a function of either just distance or just reach, and a reach is defined as the distance between 2 receiver locations. Whereas models M2 and M3 incorporated our flame data as covariates within those models. And so M2, our spacefaring model, modeled survival as a function of distinct regions that varied spatially. So with those regions defined by the cluster analysis on the mean data set, whereas M3, our time varying model, modeled survival as a function of distinct regions that varied temporally, with those regions defined by the cluster analysis on the CV data set. And so in comparing our 4 models, the spacefaring model M2 was selected by AIC as the best representation of spatial differences in survival. So I'm just going to focus on results from this model today. So we were able to estimate 2 major mortality sinks along our transect, one in the restoration area, highlighted in yellow here at C1, and then the next in the main stem central delta, highlighted in blue here at C6. And for reference, this is just around the Port of Stockton region. So for the sake of time, I'm just going to talk about environmental conditions in relation to survival between these 2 regions. So moving to the left, the top 2 line plots on the left side show survival across the 6 different regions. Those regions, again, shown spatially here on the right, with S10 on the y-axis, which stands for survival standardized per 10 kilometers, and then our 6 regions on the y-axis, or x-axis, excuse me. And then below, we have box and whisker plots of our 8 different water chemistry variables to show mean distribution of each variable across the 6 regions. And to clarify, these 2 survival graphs are identical plots and just placed here for easy comparison between survival and water chemistry values. So while we estimated similar low survival between both the restoration area and the main stem central delta, we did observe high variation in environmental conditions between these 2 regions. So I'm going to go through a couple of those. Starting with chlorophyll, we observed much higher values of chlorophyll A in the restoration area, here highlighted in yellow, compared to this main stem central delta, here highlighted in blue, as well as higher levels of dissolved organic matter in the restoration area compared to the delta. So these results may indicate differences in food resource availability between these 2 regions. Higher levels of chlorophyll A and organic matter may reflect higher levels of primary productivity and subsequently secondary production, which such as zooplankton, which are really important food resources for juvenile salmon. And low levels of these 2 variables may indicate food resource limitations here in the central delta. Additionally, we saw much higher levels of turbidity in the restoration area compared to the central delta, which could have really important implications for predation susceptibility in these 2 habitats. So previous studies have been able to show that elevated turbidity levels can decrease the visual field of fish, which may aid juvenile salmon in predator avoidance, as well as decrease predator encounter rates by increasing outmigration rate. And so higher levels of turbidity here in the restoration area may be providing salmon with additional forms of protection and cover that they're just not experiencing in the really clear water of the central delta. Oh, furthermore, our turbidity was accounted for a small but statistically significant portion of the variation in our data set, which just highlights turbidity as potentially a key variable in describing variation in survival across our transect. We also saw differences in dissolved oxygen between the 2 regions, with lower DO in the restoration area compared to the central delta. So overall, these results were a bit surprising, as we expected regions with similar survival to also reflect similar environmental conditions, but the only common factor that we observed between these 2 regions of low survival was with temperature, which was also a variable that accounted for a significant portion of the variation according to PCA. And so while these temperatures, which were just under 16 degrees Celsius, while they may not have been lethal, they could have had sublethal effects on outmigrating salmon, as well as impacting biotic factors in these 2 regions, such as pathogen loads and disease. And so we actually conducted a concurrent physiology study, which was led by PhD student Leah Mullinger, who caged juvenile salmon throughout different regions of the San Joaquin River to get a better understanding of sublethal stress responses in relation to both physical environmental factors as well as biotic factors like pathogens and disease. So we hope to see those results available next year. And in addition to this, temperature can also have really strong influences on the overall ecology of a region, particularly in relation to predators, including influencing predator body sizes, distribution and abundance, as well as metabolic demands. And so these results really emphasize the importance of managing habitat for temperature, which is supported by extensive literature on the impacts of temperature on salmon physiology and behavior, as well as predator bioenergetics. However, we do not think that temperature was the only factor driving low survival between these two regions, as the differences in survival between the restoration area in the central delta and then the intervening regions here in purple, which were much greater than expected from the differences in temperature. So in other words, the 3-degree difference in temperature accounting for a 33% difference in survival probability seems unlikely, which just indicates to us that there are most likely additional factors driving the mortality that we observed in these environments. So in the central delta, we think that warmer temperatures along with food resource limitations and high exposure to predators are likely factors working to shape the survival that we observed through this region. And while the restoration area was generally more reflective of higher quality habitat and higher habitat complexity, indicated by elevated levels of chlorophyll and organic matter, as well as higher levels of turbidity, warmer temperatures in this region could be having strong impacts on predator distributions and abundances. But another factor that we need to take into consideration is the fact that fish were released in this region here in the restoration area, and that's the mortality that we observed could in part be due to stress from trucking and handling prior to release, as well as simply hatchery fish having to acclimate to a wild environment for the first time in this region. And so in the future, we really want to take this acclimation period into account by releasing fish further upstream of the first region of interest, especially when we are comparing environmental conditions in that region to survival. Okay, so now that I've discussed survival in relation to habitat conditions along our transect, I'm going to talk a little bit about routing and survival through the remainder of the delta to the ocean, unrelated to habitat conditions, as we did not continue our environmental sampling through this region. So starting at the head of Old River, located here on the map and closer up here, which was the first bifurcated junction that fish came into contact with during their outmigration in 2019. We estimated 41% of fish to deviate from the main stem San Joaquin and enter into Old River. And of those fish that became entrained here, here at the pumping facilities at E1, E2, and D1, and then salvaged and trucked across the delta for release here in the estuary. We estimated survival to Chips Island here at A15 to be 18%. And then of those fish that entered into Old River but bypassed the pumping facilities and migrated as free swimming fish through the interior delta, meaning they were not salvaged and trucked across the delta, survival to the next downstream location here at Jersey Point was actually 0%. And looking at the Turner Cut Junction, here highlighted in yellow, we estimated the majority of fish to remain within the main stem San Joaquin at this junction, with survival from the junction to Jersey Point estimated at 46%. And then of the 20% of fish that entered into Turner Cut here at F1 and migrated through the interior delta, survival to Jersey Point was 12%. From Jersey Point through the estuary to the Golden Gate Bridge, survival was estimated at 73%. Okay, I have a couple more slides. And then overall survival from the restoration area to the ocean was estimated at 5%. So in summary, our analysis of environmental gradients across 150 kilometer section of the main stem San Joaquin revealed that variation in survival was best explained by water chemistry conditions that varied over space rather than over time. And furthermore, the study found that continuous high spatial resolution data can be used to identify habitat gradients that hold more information about survival dynamics than the commonly used geographic model or models based on distance. Additionally, we estimated survival through the delta to be really low at 13%, with 10% survival for fish that migrated through the delta using the main San Joaquin route, whereas we estimated 3% survival for fish that migrated through the delta via the Old River route. And then lastly, we observed spatial patterns in mortality in the delta to be consistent with previous years, particularly this low survival in the reach encompassing Frank's tract, here highlighted in red, which was estimated at 46% in 2019, 41% in 2018, and 18% in 2017. So Frank's tract is a large flooded island in the interior delta with really altered habitat, shallow, warm water, and a home to many non-native piscivorous predators. And so this year we wanted to focus our telemetry efforts on this region. So in addition to estimating survival through the river and delta, we added additional receivers around Frank's tract to get a better understanding of entrainment and survival in this region. And I'm not going to go too much more into this because I think I'm out of time, but I hope you guys got a chance to check out Amanda Augusta's poster. She's the master student leading up this work, and we hope to have some interesting insights on predation through the delta next year. Thank you to everyone involved in this project. This was truly a collaborative effort, and I will take any questions if there's time.
Sadly, we don't have any time, Colby. So
Sorry.
I would recommend that if any, and it's okay with you, if any audience members have questions, they can just contact you directly.
Yes, please feel free to email me. I would love to continue the conversation.
Okay. Thank you, Colby.
Thank you.